Greetings, seekers of knowledge, and welcome to another journey into the vast tapestry of science. Tonight, we embark on an exploration that challenges our very understanding of life's fundamental code. We often imagine deciphering the genome as a meticulous act of reading, base by painstaking base. But what if there was another way? A method so elegant, so intuitive, it allows us to perceive the unique signature of an organism without ever reading its entire story. Settle in, perhaps let your mind drift, or find focus in the unfolding wonder. As always, if this exploration sparks a thought, a question, or simply provides a backdrop for your own quiet contemplation, please consider sharing your insights, liking, or subscribing, so we can continue these journeys together. Good evening, and welcome back to Vibing Science. Tonight, we journey into the heart of life itself, not by meticulously reading every single word of its intricate instruction manual, but by observing its unique fingerprint, its silent code, through a revolutionary lens. Prepare to immerse yourselves in a tale of ingenuity, where simplicity meets profound insight, and where the very essence of an organism can be unveiled without ever uttering a single genetic letter. For generations, the dream of understanding life's blueprint, the genome, has captivated scientists. We've sought to categorize, to identify, to unravel the evolutionary threads that connect every living thing on this planet. And for a long time, the only path we knew was through sequencing, a painstaking, often slow, and resource-intensive process of decoding the precise order of adenine, guanine, cytosine, and thymine. But what if there was another way? A faster, more elegant method to grasp the fundamental differences and similarities between organisms, without the need for that exhaustive, letter-by-letter translation? Imagine standing before a vast library of ancient texts, each one representing the complete genetic instruction set of a different species. For centuries, our approach has been to painstakingly transcribe every single character of every single book to understand its contents. But what if instead we could simply look at the unique binding patterns of the ink on the parchment, the specific way the paper folds, the very texture of the material, and from these subtle cues discern not only which book belongs to which author, but also how closely related one text is to another? This is the essence of what we call genome profiling. Pioneered in the early 1990s by the brilliant Japanese biophysicist Professor Koichi Nishigaki and his dedicated colleagues at Saitama University, genome profiling, or GP, emerged as a paradigm shift. Initially, it was called DNA profiling, but to avoid confusion with a rapidly developing forensic technique that also used that very same name, it was wisely rebranded. And with this new nomenclature, a new era in biotech began to quietly unfold. At its core, genome profiling is a masterclass in elegant simplicity. It doesn't ask us to read the entire story, but rather to observe the unique way the story's pages melt and reform under specific conditions. Think of it like this. Every double-stranded DNA molecule, with its intricate helix, has a characteristic melting point. As you gently increase the temperature, these two intertwined strands will, at a very precise moment, begin to separate, or melt, like two lovers reluctantly letting go of each other's hands. And that specific melting behavior, that precise moment of separation, is utterly unique to its sequence. It's a signature, a genetic fingerprint. The procedure itself, while revolutionary in its outcome, is remarkably straightforward, almost disarmingly so. It all begins with a process called random PCR. Now, for those unfamiliar with polymerase chain reaction, or PCR, it's essentially a molecular photocopying machine capable of making millions, even billions, of copies of specific DNA segments. But traditional PCR requires you to know what you're looking for. You need two specific primers, short DNA sequences that latch onto the beginning and end of the target region you want to amplify. It's like needing the exact start and end page numbers to photocopy a specific chapter of our genetic book. Random PCR, however, is far more adventurous. Instead of needing two precisely matched primers, it uses just one, a single, short primer, often a universal one like PFM12, which can be used for any kind of organism, from the tiniest virus to the largest whale. And here's the clever bit. It operates at a much lower annealing temperature, around 30 degrees Celsius. This relaxed environment allows for less stable template primer pairings, meaning the primer doesn't have to be a perfect match to bind. It's like letting our molecular photocopying machine randomly pick sections to copy, rather than strictly sticking to a predefined chapter. The result is a diverse collection of DNA fragments, amplified from various, often unexpected, regions of the genome. While these fragments are not intentionally designed, their generation is theoretically predictable if you know the template and primer sequences. Once we have this medley of randomly amplified DNA fragments, the next crucial step unfolds: microtemperature gradient gel electrophoresis, or TAGE. Imagine a thin, jelly-like slab, a gel, with an electric current running through it. When you load your DNA fragments onto this gel, they'll begin to migrate, with smaller fragments moving faster and further than larger ones. This is standard gel electrophoresis. But here's where the temperature gradient comes in. As these DNA fragments journey through the gel, the temperature of the gel itself is steadily increasing along its length. Now, recall our earlier discussion about DNA melting. As each double-stranded DNA fragment encounters higher temperatures within the gel, it eventually reaches its specific melting point. When a fragment denatures or melts, its structure changes, and this change significantly affects how it moves through the gel. It's like a swimmer suddenly changing their stroke in a river. Their speed and direction will shift. This sudden transition in mobility creates a distinct species-specific pattern of bands on the gel, a unique genome profile. Each band's position isn't just about its size, but also about its precise melting behavior, which is dictated by its underlying DNA sequence. It's a visual representation of the inherent stability and sequence of the DNA, without ever having to read it letter by letter. From these intricate genome profiles, we then extract what Professor Nishigaki and his team term species identification dots, or Spiddos. These Spiddos are essentially the initial melting points for each of those distinctive DNA bands. Their positions are directly determined by the DNA sequence, making them theoretically predictable. In essence, Spiddos encapsulate what's known as SIOWS, sequence inheritable information obtained without sequencing. This is the very core, the unique essence of GP technology built upon the fundamental principles of DNA melting theory. It's a brilliant conceptual leap. Instead of reading the book word for word, we're extracting key structural markers, like the unique font sizes or paragraph breaks, that still tell us a profound story about its identity. With these Spiddos in hand, we can then begin to truly compare and contrast organisms. The difference between two genomes can be quantified through a pattern similarity score, or PASS. This parameter isn't just for identifying species. It's a powerful tool for classification, allowing us to build a precise, numerical understanding of how closely related two organisms are, and even to measure the degree of mutation within a genome. It's like developing a sophisticated algorithm to compare the unique structural fingerprints of our ancient texts, and from that, determining their shared lineage or divergent evolution. The versatility of genome profiling is nothing short of astounding. From its inception, it has been successfully applied to a breathtaking array of organisms, a testament to its universal nature. Imagine identifying viruses and bacteria with unprecedented speed, or classifying fungi and protozoa with a clarity previously unattainable. Early studies on insects, fish, and plants revealed a remarkable congruence between GP-based clustering and traditional phenotype-based taxonomy. This means that the classifications derived from these silent genetic fingerprints aligned perfectly with what scientists had observed and categorized for generations, based on physical characteristics. In fact, for insects, GP has even proven to be superior to traditional sequencing-based methods like 18S rDNA sequencing, offering a more efficient and accurate classification. The vision extends even further. A GP-based genome database is currently under development, aiming to map organisms within a genome sequence space. In this conceptual landscape, the closer the similarity between organisms, the closer their spatial representation in the database. It's a revolutionary way to visualize the tree of life, not through endless strings of A's, T's, C's, and G's, but through the elegant geometry of their melting profiles. Beyond fundamental research, GP has found practical applications in areas like authenticating fungal culture collections, ensuring the purity and identity of vital biological resources. It has been used to detect irreplaceable samples, such as single-cell protists like radiolaria and foraminifera, which are often challenging to analyze with traditional methods. Even in forensics, GP has shown promise, aiding in the identification of body fluids like blood, saliva, and semen. For scientific inquiry, it has helped discover continuous mutations in body cells, distinguish the origins of leaves from different trees in an ambient environment, and even determine the family relationships of mice. Perhaps one of the most exciting applications is in the field of toxicology with the development of GPMA, the GP-based mutation assay. Here, organisms like the common bacterium Escherichia coli are exposed to potentially mutagenic agents, be they physical or chemical. By comparing the genome profiles before and after exposure, the changes in genome distance can precisely indicate the extent of DNA damage and mutation. This offers a rapid, cost-effective way to screen for environmental hazards and assess their impact on genetic integrity. In an age where next-generation sequencing often implies ever more sophisticated, complex, and expensive machinery, genome profiling stands as a powerful testament to the enduring value of simplicity and ingenious design. It offers a complementary, yet profoundly significant, approach to understanding the genetic world around us. It's a technique so straightforward that it primarily requires only a single random PCR and a micro-TG experiment, an entire process that can be completed in approximately 2 hours. The elegance lies in its universality. A single, universal primer can be used for any organism, yielding the universal parameter of Spiddos, which in turn allows us to measure genome distance across the entire spectrum of life. This means that genome information can be compactly stored and efficiently utilized in internet databases, moving away from unwieldy, raw sequence data toward highly informative, processed fingerprints. While full automation is a future goal that would further enhance its accessibility, the current system, comprising PCR, TG, an imager, and a computer, already offers an inexpensive and versatile toolkit, with components that can be used for multiple purposes independently. So, as we contemplate the vast, intricate tapestry of life, remember that sometimes the deepest insights aren't gained by reading every single thread, but by understanding the unique way those threads intertwine and respond to the gentle touch of the universe. Genome profiling invites us to listen to the silent code, to perceive the hidden patterns, and to appreciate the profound stories etched into the very melting points of DNA. We'll take a brief pause here. When we return, we'll delve deeper into the elegant dance of DNA within this process, exploring the mechanics of random amplification and temperature gradient electrophoresis in greater detail. Stay with us. In our exploration of life's intricate tapestry, we now delve deeper into the elegant dance of DNA itself, moving from the conceptual understanding of genome profiling to the ingenious mechanics that allow us to glimpse its secrets without the laborious task of full sequencing. This is where the magic of random amplification and temperature gradient electrophoresis truly begins to unfold, painting a picture of unparalleled efficiency in the realm of biological discovery. Imagine, for a moment, the vast, undulating ribbon of a genome. It's a colossal blueprint, thousands, even millions of pages long. Traditional sequencing methods are akin to reading every single word on every single page, a monumental undertaking that, while incredibly precise, is often time-consuming and resource-intensive. But what if there was a way to understand the essence of that blueprint, to grasp its unique signature, without needing to decipher every single letter? This is the core brilliance of the method we're discussing. Pioneered by the insightful mind of Japanese biophysicist Professor Koichi Nishigaki and his dedicated colleagues at Saitama University in 1990, this revolutionary approach was initially termed DNA profiling. However, to avoid confusion with emerging forensic technologies bearing the same name, it was wisely rebranded as genome profiling. This subtle shift in terminology marks a profound conceptual difference, signaling an investigation into the entire genetic landscape, not just specific markers. At the heart of this technique lies a seemingly simple yet profoundly clever trick: random PCR, or random polymerase chain reaction. Now, for those familiar with the standard PCR, you'll know it's a process of amplifying specific segments of DNA, much like making millions of copies of a particular sentence from our blueprint. It typically requires two specific primers, short DNA sequences that act like bookmarks, guiding the copying machinery to the exact start and end points of the desired segment. But here's where random PCR diverges, with a stroke of genius. Instead of two precisely designed primers, random PCR employs only a single, short primer. And crucially, it operates at a significantly lower annealing temperature, around 30 degrees Celsius, compared to the much higher temperatures of conventional PCR. What does this seemingly small adjustment achieve? It allows for less stable, more opportunistic template primer hybrid structures to form. Think of it like this. Instead of needing perfect, tightly bound bookmarks, random PCR allows for a more relaxed, anywhere near here is good enough approach. This seemingly less precise method, surprisingly, leads to a highly predictable and species-specific outcome. This single, short primer, remarkably, can be a universal key. A specific primer, PFM12, a mere 12 letters long, has been found to work across a breathtaking spectrum of life, from the smallest virus to the largest whale. This universality is a critical advantage, meaning you don't need prior knowledge of an organism's specific gene sequences to begin profiling its genome. You don't need to know what you're looking for to find its unique signature. The beauty of random PCR is that even though the amplification appears random in its initial target selection, the resulting DNA fragments are, in fact, theoretically predictable based on the template and primer sequences. It's like scattering a handful of magnets over a metal sheet. While their initial landing points might seem random, their final positions, once they've settled and aligned, are governed by predictable physical laws. These randomly amplified fragments are not just noise. They carry a silent code, a whisper of the organism's unique genetic identity. Once these numerous, randomly amplified DNA fragments are generated, they embark on the second, equally crucial stage of the process: microtemperature gradient gel electrophoresis, or TAGE. Imagine a long, thin gel, similar to the jelly-like substance used in many biological labs to separate molecules. But this gel has a special property. It has a carefully controlled temperature gradient running along its length. One end is cooler, the other end progressively warmer. When the randomly amplified DNA fragments are loaded onto this gel and an electric current is applied, they begin to migrate. Smaller fragments move faster, while larger fragments move slower. This is standard gel electrophoresis. However, the temperature gradient adds another layer of exquisite separation. Double-stranded DNA, the form in which our genetic material usually exists, is quite stable. But as the temperature increases along the gel, each individual DNA fragment reaches a specific melting temperature. At this critical point, the two strands of the DNA helix begin to separate or denature. Crucially, the exact temperature at which a DNA fragment denatures is highly dependent on its unique sequence of nucleotides. the A's, T's, C's, and G's that make up the genetic code. A fragment rich in GC pairs, for example, will be more stable and melt at a higher temperature than a fragment rich in AT pairs. This melting causes a sudden, distinct change in the fragment's mobility through the gel. It's like a swimmer suddenly hitting warm water and changing their stroke. The result of this elegant interplay of size and melting behavior is a distinctive mobility pattern on the gel, a series of bands, each corresponding to a specific DNA fragment, appearing at precise positions determined by its unique sequence and its melting characteristics. This pattern, much like a barcode, is species-specific. It is the genome profile itself. From these intricate patterns, specific markers known as species identification dots, or Spiddos, are extracted. These Spiddos are essentially the initial melting points for each distinct DNA band on the gel. Their positions are not arbitrary. They are directly determined by the DNA sequence, by the very arrangement of letters in the genetic blueprint. This means that, theoretically, these Spiddos are predictable. They carry a unique kind of information, dubbed SIOWS, sequence-inherent information obtained without sequencing. It's an ingenious way to distill the essence of a DNA sequence without having to laboriously read every single base pair. Imagine trying to recognize a friend in a crowd. You don't need to know their entire life story. A few key features, their gait, their hair color, their favorite shirt, might be enough. Spiddos are those key features, providing enough information to identify and distinguish organisms with remarkable accuracy. But the utility of this approach extends beyond mere identification. With Spiddo information in hand, we can now quantify the differences between two genomes. This is achieved through a pattern similarity score, or PASS. Think of it as a mathematical measure of how alike two genome profiles are. The closer the patterns, the higher the similarity score, and the more closely related the organisms. This parameter has proven invaluable for species identification, classification, and even for measuring the subtle degrees of genetic mutation. The entire process from the random PCR to the TAGE experiment can be accomplished in approximately 2 hours. Contrast this with the days, or even weeks, required for full genomic sequencing. This ultra-high performance, combined with its ability to work without prior sequence knowledge, makes genome profiling a truly powerful and accessible tool for a vast array of biological questions. It's a testament to human ingenuity, finding a shortcut to understanding the profound complexities of life by observing the elegant dance of DNA and deciphering its silent, yet unmistakable code. This method isn't just a technological marvel. It's a new lens through which we can perceive the interconnectedness and diversity of life on Earth, and perhaps beyond. Now, we journey beyond the mere mechanics, into the profound implications of this silent code. We've laid the groundwork for how genome profiling works, how those tiny DNA fragments are amplified and then paraded through a temperature gradient, revealing their unique melting points. But what do we do with this information? What do these patterns, these silent echoes of a genome, truly tell us? This brings us to a remarkable concept, the Spiddos. Imagine these as individual identity markers, specific points on that electrophoretic gel where a DNA band first begins to unravel or melt. Each Spiddo is a distinct fingerprint, determined by the underlying DNA sequence. This is the truly elegant part. We are extracting information inherently embedded within the DNA sequence, what Professor Nishigaki and his team termed sequence-inherited information obtained without sequencing, or SIOWS. It's like understanding a complex melody not by reading every single note, but by recognizing the unique way certain phrases begin and end. Each Spiddo holds a secret, a piece of the genomic puzzle, all without the arduous and time-consuming process of deciphering every single letter in the genetic code. From these Spiddos, we can then construct a powerful metric, the genome distance. Think of it as a measure of how similar or dissimilar two genomes are. If two organisms have very similar Spiddo patterns, their genome distance is small, indicating a close evolutionary relationship. If the patterns are vastly different, the distance is large, signifying a more distant connection. This isn't just a theoretical exercise. This parameter, the pattern similarity score or PASS, has been astonishingly successful in identifying species, classifying organisms, and even quantifying the subtle shifts of mutation. It's a profound shortcut to understanding the intricate relationships woven into the fabric of life. The genius of genome profiling lies in its ability to reveal these deep connections without the need for prior knowledge of an organism's genetic makeup. Traditional PCR, for instance, demands that you already know specific sequences to design your primers. But with random PCR and its universal primer, like the now famous PFM12, it's a completely open-ended exploration. You don't need a map, you just need to begin the journey. This allows us to venture into the unknown, to profile organisms about which we know very little, and still gain profound insights into their identity and evolutionary position. The applications of this technology are vast and incredibly diverse, spanning the entire spectrum of life. From the simplest viruses and bacteria, through fungi and protozoa, to complex insects, fish, and even large animals and plants, genome profiling has proven its mettle. Early studies beautifully demonstrated the congruence between classifications derived from genome profiling and those established through classical, phenotype-based taxonomy. Imagine seeing the same family trees emerge, independently, from two entirely different methods, one based on observable traits, the other on the silent code within. In some cases, genome profiling has even outshone traditional sequencing. For instance, classifying insects, with their bewildering diversity, can often be more straightforward and efficient using GP than with conventional approaches like 18S ribosomal DNA sequencing. This is because GP offers a holistic view, a fingerprint of the entire genome, rather than focusing on a single, albeit important gene. The vision for genome profiling extends even further, a universal genome database. Imagine a sprawling digital map where every organism, from the smallest microbe to the largest whale, is precisely located within a genome sequence space. The closer their similarity, the closer their position on this map. This isn't just an abstract idea. It's a living project, constantly growing, providing a new lens through which to understand biodiversity and evolution. Beyond broad classification, GP has found its niche in highly specialized applications. It's been used to authenticate precious fungal culture collections, ensuring that what scientists believe they are studying is indeed what it is. It can detect irreplaceable samples, such as those incredibly delicate single-celled protists, like radiolaria and foraminifera, whose morphology alone might not fully reveal their identity. In forensic science, it offers a quick and robust method for analyzing body fluids, providing crucial links in investigations. For fundamental scientific inquiry, genome profiling is a powerful tool. It has been used to detect the continuous mutation of body cells, a subtle dance of genetic change within our own tissues. It can discriminate the precise origin of a leaf, telling us which tree it fell from in a forest. It can even determine the intricate family relationships of mice, mapping their lineage with unprecedented clarity. One particularly significant application is the Genome Profiling-Based Mutation Assay, or GPMA. By exposing a test organism, like the bacterium Escherichia coli, to potential mutagens, whether they be physical agents like radiation or various chemicals, scientists can then use genome profiling to detect the subtle sequence changes, the shifts in the Spiddo patterns. This provides a rapid and accurate way to identify substances that can damage DNA, offering crucial insights for environmental protection and human health. In essence, genome profiling is a unique and wonderfully efficient technique. It offers us useful information without sequencing, those elusive SIOWs, expressed as Spiddos. It's a testament to scientific ingenuity, a method that is elegantly simple in its execution, requiring only a single random PCR and a micro-TG experiment, a process that can often be completed in a mere 2 hours. This simplicity, combined with its universality, the ability to use the same primer for any organism, makes it an unparalleled tool for measuring genome distance and, in turn, understanding the vast tapestry of life. While the age of next-generation sequencing often emphasizes high-throughput, complex procedures, the genius of genome profiling lies in its complementary role. It's a testament to the power of elegance and efficiency. It doesn't replace sequencing. Rather, it offers a swift, cost-effective, and remarkably insightful pathway to understanding the fundamental blueprints of life, often revealing truths hidden in plain sight, without the need to read every single word. It's a silent, profound conversation with the very essence of existence. From the silent whisper of a virus to the majestic song of a whale, the principles we've been exploring, the elegant dance of DNA, melting, and profiling, find a breathtaking universality. This isn't just a fascinating laboratory trick. It's a profound key, unlocking the secrets of life across the vast tapestry of biological existence, often with astonishing speed and simplicity. Imagine, for a moment, the sheer diversity of life on Earth. A microscopic bacterium, an ancient redwood, a flickering firefly, or a human being, all fundamentally built upon the same exquisite molecule, DNA. Yet, traditionally, understanding the intricate relationships between these organisms, or even simply identifying them, has been a monumental undertaking, often requiring the painstaking letter-by-letter deciphering of genetic codes. But the method of genome profiling, or GP, offers a different path, a more intuitive approach, like recognizing a face without needing to read a full biography. It has been applied to an incredibly broad spectrum of life forms, spanning the entire tree of life, from the very simplest of pathogens to the most complex multicellular organisms. Think about that for a moment. A single, universal technique capable of shedding light on the genetic makeup of anything alive. Early on, scientists like Kaduka and their collaborators made a remarkable discovery. When they used GP to classify various insects, fish, and plants, their results, the patterns emerging from those temperature gradient gels, aligned almost perfectly with the established classical taxonomies. These traditional classifications, built over centuries through careful observation of physical traits and behaviors, found a powerful, independent confirmation at the molecular level. It was as if GP was speaking the same ancient language of biological relatedness, but with a new, remarkably efficient dialect. In fact, for certain organisms like insects, GP proved to be even more effective and straightforward than conventional sequencing-based methods. Consider the 18S rDNA sequencing, a common tool in molecular taxonomy. While powerful, it often requires specific primers and a deeper dive into the sequence itself. GP, with its universal primer and focus on melting patterns, cuts directly to the chase, revealing the inherent genetic signatures that distinguish one species from another. It's like being able to identify a piece of music by its unique rhythm and melody, rather than having to read every single note on the score. This unparalleled efficacy has even led to the ambitious proposal of a GP-based genome database. Imagine a vast, interconnected digital space where every organism, from the simplest to the most complex, is positioned based on its genetic similarity. The closer their evolutionary kinship, the closer their distance in this genomic landscape. This isn't just a theoretical construct. It's an ongoing endeavor, building a new kind of biological map, guided not by arbitrary classifications, but by the fundamental patterns of DNA itself. The applications extend far beyond mere classification. In the practical world, GP has become an invaluable tool for ensuring the authenticity of precious fungal culture collections, protecting against misidentification or contamination. It has also proven vital in the study of irreplaceable samples, such as the single-celled protists, those ancient and often enigmatic inhabitants of our oceans, like radiolaria and foraminifera, whose delicate forms are often difficult to study by other means. And in the field of forensics, GP can even be used to analyze body fluids, blood, saliva, semen, providing crucial information for identification and investigation, all without the time-consuming process of full genome sequencing. For the pure pursuit of scientific understanding, GP has opened new avenues of exploration. It has been used to detect the continuous, subtle mutations occurring within the body cells of an organism, offering insights into aging and disease processes. It can even discriminate the precise origin of leaves from different trees in an ambient environment, a testament to its fine-grained discriminatory power. And perhaps most endearingly, it has helped determine the intricate family relationships within groups of mice, painting a clearer picture of their genetic lineage. One of the most profound extensions of GP is its application in detecting mutagenic chemicals, those substances that can alter our DNA. This innovative technology is known as GPMA, the GP-based mutation assay. Here, a test organism, often a bacterium like Escherichia coli, is exposed to suspected mutagens, be they physical agents like radiation or various chemical compounds. By then analyzing the resulting changes in their genome profiles, specifically through the calculation of genome distance, scientists can quickly and efficiently determine if these agents are indeed causing alterations to the genetic sequence. It's a rapid, powerful screen for potential environmental hazards, a watchful guardian against the unseen forces that can reshape life's fundamental blueprint. In essence, genome profiling stands as a unique testament to the ingenuity of scientific inquiry. It's a technique that provides deeply useful information, what we call SIOWS, or sequence-inherent information obtained without sequencing, information expressed vividly through those species identification dots, or Spiddos. And it does all of this without demanding the exhaustive and often time-intensive process of full DNA sequencing. What makes GP truly remarkable is its elegant simplicity. It can be performed with just a basic random PCR and TGG setup, requiring only a single, universally applicable primer. This universality means the same primer can be used for any organism, leading to the acquisition of those critical Spiddos, which in turn allow us to precisely measure genome distance between any two life forms. It's a universal Rosetta Stone, unlocking the language of genetic relatedness across all species. Imagine the potential. This compact, powerful genome information, encoded in Spiddos, could be stored and accessed efficiently in an internet database, creating a truly global and accessible library of life's genetic patterns. While there are still steps that could be automated to make the entire GP procedure even more seamless, currently, there's a small manual step involved. The existing system offers a powerful balance. The components themselves, the PCR machine, the TGG apparatus, the imager, the computer, are all inexpensive and versatile, serving multiple purposes in a laboratory. In an age increasingly dominated by next-generation sequencing, with its sophisticated and sometimes overwhelmingly complex procedures, the elegant simplicity of genome profiling offers a vital complement. It reminds us that sometimes the most profound insights don't require the most intricate tools, but rather a clever and insightful way of asking the right questions. It's a powerful illustration that understanding the vast, silent code of life doesn't always demand reading every single letter, but sometimes simply observing the beautiful, inherent patterns it creates. And in that observation, we find a new, awe-inspiring perspective on the interconnectedness of all living things. And so, our journey through the intriguing world of genome profiling concludes, leaving us with a renewed appreciation for the ingenuity of scientific discovery. We've seen how the subtle dance of DNA, influenced by temperature and universal primers, can reveal profound truths about identity, ancestry, and even the unseen impacts of our world. It's a testament to the fact that even in an age of complex sequencing, simplicity can hold unparalleled power. I sincerely hope this deep dive has offered you a fresh perspective, perhaps even a moment of quiet wonder. As always, your thoughts are invaluable. Do let me know in the comments what you found most fascinating or what other cosmic or quantum mysteries you'd like us to explore next. Until our next convergence, keep questioning, keep exploring, and keep vibing with science.